Hello, this is Professor Beida back again with another biology video. Today we will be doing chapter 19 and 27, which is viruses and prokaryotes. So I will start with the viruses, and just let me warn you that this chapter has a lot of specific details that we don't need to know. So basically, just get the overall ideas, and I will talk about what's important to know. Compared with eukaryotes and even prokaryotic cells, most viruses are much smaller and simpler in structure. A virus is infectious particles consisting of little more than genes packaged into a protein coat. Are viruses living or non-living? So early on, they were considered biological chemicals. So the Latin root for virus means poison. Viruses can cause a wide variety of diseases, so researchers in late 1800s saw a parallel with bacteria and proposed that viruses were the simplest of living forms. However, viruses cannot reproduce or carry out metabolic activities outside the host cell. Most biologists would probably agree that viruses are not alive but instead exist in a shady area between a life form and chemicals. The simple phrase used by two researchers describes them aptly. Viruses lead a kind of borrowed life. The tiniest viruses are only 20 nanometer in diameter, smaller than a ribosome. Examining the structure of a virus more closely reveals that it is infectious particles consisting of one or more molecules of nucleic acid, enclosed in a protein coat and for some viruses surrounded by a membranous envelope. The simple structure of viruses make them a useful biological system. To a large extent, molecular biology was born in laboratories of biologists studying viruses that infect bacteria. Experiments using these viruses provided evidence that genes are made up of nucleic acid, and they were critical in working out of the molecular mechanism of fundamental processes of DNA replications, transcriptions, and translation. We usually think of genes as being made of double-stranded DNA, but many viruses defy this convention. Their genomes may consist of double-stranded DNA or single-stranded DNA, double-stranded RNA or single-stranded RNA, depending on the type of virus. A virus called a DNA virus or an RNA virus based on the kind of nucleic acid that makes up its genome. The genome is usually organized as a single linear or circular molecule of nucleic acid. Although the genome of some viruses consists of multiple molecules of nucleic acid, the smallest viruses now have only 3 genes in their genome, while the largest have several hundred to 2,000. For comparison, bacterial genome contain about 200 to a few thousands of genes. The protein shell enclosing the viral genome is called a capsid. Depending on the type of virus, the capsid may be rod-shaped, polyhedral, or more complex in shape. Capsids are built from a large number of protein subunits called capsomeres. But the number of different kinds of protein in capsid is usually small. Tobacco mosaic virus has a rigid rod-shaped capsid made from over 1,000 molecules of a single type of protein arranged in helix. Rod-shaped viruses are commonly called helical viruses for this reason. And this is tobacco mosaic virus. I mentioned before that has a rigid rod-shaped capsid made from over 1,000 molecules of single type of protein that arranged in helical or rod shape virus. And we call them helical viruses for this reason. And again, it has a helical capsid surrounding a single helical RNA molecule with the overall shape of rigid rod. In part B, we have an adenovirus. Adenovirus have an icosahedral capsid that encloses the DNA with the protein spike at each corner. The capsid made up of capsomeres contain double-stranded DNA. And in part C, we have influenza virus, have an outer envelope studded with glycoprotein, and that contain 8 double helical RNA protein complexes. In part D, we have bacteriophage. Bacteriophage has a complex capsid consisting of an icosahedral head and a tail apparatus, and consisting of the tail sheath. This one is slender, and tail fibers. Those are tail fibers. The double-stranded DNA is housed in the head. Some viruses have accessory structure that help them infect their hosts. For instance, a membrane envelope surrounded the capsid of influenza viruses and many other viruses found in animals. The viral envelope, which are derived from the membrane of the host cells, contain host cell phospholipids and membrane protein. They also contain protein and glycoprotein of viral origin. Remember that glycoproteins are proteins with carbohydrates covalently attached with each other. Viruses lack metabolic enzymes and equipment for making proteins, such as ribosomes. They are obligate intracellular parasites. In other words, they can replicate only within a host cell. Each particular virus can infect cells of only a limited number of host species. We call them the host range of the virus. This host specificity results from the evolution of recognition system by the virus. Viruses usually identify host cells by, let's say, by handshake, fit between viral surface protein and the specific receptor molecule on the outside of the cell. Of course, those receptor proteins in the cell membrane. Such receptor molecules are most often proteins that carry out necessary function for the host cell and have been adapted by viruses as a portal of entry. Some viruses have broad host ranges, but other viruses have host ranges so narrow that they infect only a single species. Measles viruses, for example, can infect only human. A viral infection begins when a viral binds to a host cell and the viral genome makes its way inside. The mechanism of genome entry depends on the type of virus and the type of host cell. Other viruses are taken up by endocytosis, or in the case of enveloped viruses, by fusion of the viral envelope with the host's plasma membrane. Once the viral genome is inside, the proteins it encodes can commandeer the host, reprogramming the cell to copy the viral genome and manufacture viral proteins. The host provides the nucleotide for making viral nucleic acids as well as enzymes, ribosomes, tRNA, mRNA, amino acids, ATP, and other components needed for making the viral proteins. After the viral nucleic acid molecules and capsomeres are produced, they spontaneously self-assemble into new virus. This is a simplified viral replicative cycle. A virus is an intracellular parasite that uses the equipment and the small molecules of its host cell to replicate. In this simplest of viral cycle, the parasite is a DNA viruses with a capsid and consisting of a single type of protein which enter the cell. Um, Here, the virus enter the first step enters the cell and is uncoated, releasing its viral DNA and capsid protein. In number two, the host enzymes replicate the viral genome and inside the cell, while mean host enzymes inside the cell transcribe the viral genome into viral mRNA, which host ribosomes use to make more capsid proteins, and then viral genomes and capsid proteins self-assemble into new virus particles which exit the cell. And now I will let you watch this video.